In this video, we will discuss about this FAS-DNR modified T cells in the autoimmune disease. So they check their construct with the dominant negative receptors of FAS in the in vivo studies that in which a mice is infected with this autoimmune disease. So how they did their experiment? They took this mice, the B6 wild type mice, and inject them with this virus, and after one day they infuse this PMEL either with empty vectors or the vectors that does not have this FAS intercellular signaling uh, domain in the CAR construct. And for the two, they also inject this IL-2 for the 3 days after the injections of this CAR construct. And then after 200 days, they check the T cells lymphocytes in the spleen. So in the wild type control, where they don't use anything, don't transduce them with any tumors, they do not see too much signals for this B6 IPR, whereas the cells that are transduced with these tumors but are not transduced with the CAR cells shows a high tumors in the spleen, that is around 21.1% after 6 months. And to their surprise, they do not see any tumors formations even in the empty vectors, that is mice that is transduced with the empty vectors. And for the case where mice is transduced with empty vectors, and also this FAS, and for the case where mice is transduced with a CAR construct where FAS intercellular domain is truncated, here also they do not see any tumor populations or B6 IPR positive populations. And these things they have quantified in the panel C, where we can see that in the wild type they do not see any CD3 and B220 plus positive populations, and but for this B6 IPR they see a high CD3 V220 population. This is a positive control. And for the empty construct treated mice, they do not see any significant increase in the populations. And for the case where CAR constructor does not have FAS intercellular signaling domain, also does not show any any significant increase in the CD3 plus and B220 plus positive populations. And thereafter, at this point, they do not see a difference in the modified T cells, either in the case where CAR FAS is intercellular FAS is truncated, or the mice is treated with empty vectors. So they continued their experiment till 6 months, around 200 days, and then they are able to see some changes. Right? In the case of empty construct, they see around this population, some populations, that is thigh 1.1 positives, and which is the markers for a CAR construct. And out of that, they can see that around 220 B220 positive cells are around 100%, almost negligible. Whereas in the case where FAS signaling pathways has been truncated, they see that around 98.1% of those thigh 1.1 positive cells shows negative for the the B220 negative cells. So this might indicates that cells that are transduced with the or we can say that the CAR T cells that are modified with this FAS intercellular domains truncations does not get affected with the does not get affected with the tumor cells that is present in the mice. And the quantifications of this panel D's results are shown in this panel E, where we can see that the thigh positive cells after per spleen in the spleen is almost negligible, whereas in the case where CAR construct is where the FAS intercellular signaling domains is truncated, they see a high positive populations for the thigh 1.1 and PMEL 1 cells per spleen. This also indicates the one of the but this also indicates their previous hypothesis that the cells that have intercellular signaling domains get activated in the activated T cells, and thereby it will cause the apoptosis of the T cells. This is the reason. This could be the reasons why this empty construct cells has very low Thi 1.1 positive cells. Most of those cells might be triggered apoptosis by FAS signalings, whereas this is not the case, where FAS intercellular domain signaling has been truncated. So that's all for this video. I hope you understood how they check their FAS dominant negative receptors, modified T cells in the autoimmune disease, the in vivo studies. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.